We here at Diagnomics are committed to ushering in the next generation of healthcare based around personalized medicine. Today we'd like to introduce our services based upon individual genetic information called the Personal Genome Annotation System, or PGAS for short. This system is based upon your genetic information, i.e. your genome. The following tutorial will show you how to use our system. This service starts with sequencing your genome, assembly and alignment, then lastly variant identification and annotation from next-generation sequencing. Our system takes advantage of research from various sources including public databases, literature, and other scientific work to analyze your genetic information. This system is constantly updated and is streamlined to offer summary-level statistics down to the variant level. Variant here is defined as any variation, whether it be a SNP, insertion, deletion, or copy number variation, or any other structural variation. A major distinction we cannot stress enough between our system and other current offerings is that PGAS is based upon genome sequencing data. This is unlike other services which only use SNP genotyping data. This allows our system to analyze all known variants from SNPs, insertions, deletions, copy number variants, and any other unknown variant discovered in the future, unlike the other services which only rely on fixed SNP data. Keep in mind that we can always update our system with new information and reanalyze your sequencing data. This allows our system to remain cutting edge and stay ahead of other current offerings.
Our personal annotation system is a web-based tool that can be accessed from your computer, your smartphone, or any internet-enabled device. In addition, there are summary and raw reports if you choose to print a hard copy. To access your information, go to our Personal Genome Annotation site and log in. This is the GAS homepage. Here you can load, save, reset, or otherwise manipulate sessions. Now let's walk through an example of a few interesting protocols. First, we will select one sample. In this case, I will be selecting sample 9. The variant count number tells you how many differences you have compared to the reference genome generated by the Human Genome Project. In this case, this individual has roughly 4.6 million differences than the Human Genome Project. Just a reminder, variants are any differences and in this case include SNPs, insertions, deletions, or copy number variants. Perhaps you're not interested in all the variants. Now that a sample is selected, you can apply various different filters to reduce the number of results. As we apply filters, the variant count number will reduce. These filters fall under 5 general categories as you can see from the main headings on the left column. They are carrier effect, coordinate, genotype, variant, and dbSNP. We do not have the time to explain them all today, but we will show you a few common ways to filter. One common way to filter is by specific locations within the human genome. The human genome has 23 chromosomes, but in this case, let's say that you're only interested in variants on the 12th chromosome. We can click Chromosome, change the value by selecting the widget on this filter. In this case, I'm interested in chromosome 12. Apply the change, and now our filter showing only chromosome 12 results is applied to the sample. You can click Run Filter at any time to see your results. This may take up to a minute or two depending upon server load and the filters applied. As you can see, our variant count number is reduced from roughly 4.6 million down to 213,000. There are other ways to filter, and you can run multiple filters on a given sample. In this case, let's say I'm also interested in only those variants that offer a change. So I can apply the change effect filter, as you see here, and click Run Filter again to see my new results. We would expect the filter result number to be less than previously. As you can see here, we only have 217 instead of 213,000. Now that we have some filters applied, there are many ways to view your results. In this case, you can click on any of the results to see your specific filtered results. In this case, I'm going to click on all, the 4.6 million. What you see here is currently the body map view. This allows you to select different areas of the body and see related variations. In my example, this individual has an increased risk of baldness, straighter hair, and typical curliness. You can see this described in each row of the table: phenotype, the associated SNP, genotype, and result. That is, this single change in your DNA can change your health. In this case, the individual, according to known research, has an increased risk of baldness. Again, we want to remind you that genetics only partially explains, if at all, the expressed phenotypes. That is, not everything is genetically determined, and our current understanding of our genetic information can only explain a portion, if any, of our health. Now let's click on the face region. One interesting fact is what's called alcohol flush reaction. You know when you drink with other people and some of them turn bright red or have rosy cheeks? This can be determined genetically. In this case, our sample does not have a mutation and thus does not develop a flush reaction to alcohol. That is, they do not turn red. Now on to a few more items pertaining to health, then say eye color. In the face region, high myopia or a disease of the eyes can be explained genetically. Other interesting health concerns such as your smoking behavior and sugar consumption can also be addressed genetically, as seen here in this region. Drugs and how we respond to them is one of the cornerstones of personalized medicine. Here I'm going to click on drug metabolism. Drugs specifically interact with different systems of our body, and thus how our body responds to certain drugs can to some degree be explained with our current understanding of genetics. For example, this individual has a normal rate of remission after being on antidepressants such as Paxil. There is another way to view this information that is designed for risk assessment. We'll talk about it more later. Taking a step back, we're still in the overview view. This lets you see your genetic information via the body map system, variation, which is done by location, phenotype, which is also done by location, and statistics. Variation shows you the variance based upon chromosomal location and can be zoomed in. As you can see here, each one of these lines represents a variant on that particular chromosome. Phenotype colors these variants along the chromosomes, whether it's associated with rare or common diseases, drug response, or physical traits. Statistics, on the other hand, gives you a whole genome view of your variants. This allows researchers to see the whole genome view as well as single variant details. Now let's view our diseases and traits. This view has 4 tabs: rare disease, common disease, drug response, and physical traits. Each view has a tip box at the top which explains how the results are formatted. For this individual, they have 0 genetic variants that could cause rare genetic disease according to a screening of a genetic database called OMIM. Let's click on common disease for now. Here again is a tip box which helps explain how the results are shown. In general, you can briefly view the information based upon colors, which show you high, normal, and low risk. For those of you that want numbers, you can see a relative risk as well as a prevalence rate. Prevalence is the total number of affected individuals out of a population, so a 10% prevalence rate means that 1 in 10 people have this disease. Again, a reminder that because you are genetically predisposed, that is, you have a higher chance of having the disease, it does not mean you will automatically develop the disease. Also, if you are at a reduced risk, you may still develop the disease. For example, this individual is roughly 4 times more likely to develop schizophrenia, and 2 times more likely to develop asthma than the average individual. Keep in mind that the chance is still relatively low, though, roughly 1 in 40 for schizophrenia and 1 in 9 for asthma. So why do we care about high-risk diseases? Simply so that we can avoid lifestyles that may further increase this risk and thus maintain good health. If you knew you had a higher chance to suffer from high cholesterol, you would consult your doctor and control your diet. Just knowing that you are at risk allows you to take preemptive measures to ensure your well-being. Another great example is diabetes. Type 2 diabetes, the common type, is currently attributed primarily to lifestyle such as high sugar diets. Therefore, if you know you are at higher risk to develop diabetes, you may change your lifestyle accordingly. For further information on each disease, consult your doctor or another verified medical source. Let's revisit drug response. This view is identical to the rare and common disease tabs. Here you can see conditions related to drugs and you. In this example, this sample has a low chance of developing alcohol dependence, has higher chances of developing an allergy to penicillin, and responds normally to antidepressants. Physical traits. This section shows you physical traits that can be explained genetically to some degree. For example, this person is likely to have freckles and is probably taller than average. Freckles and height. We can also trace our ancestry genetically. The world map view shows you how the individual relates to known world populations. This individual is genetically from Eastern Asia, as we can see by the results here. You can see that most of his variants and trace back is from Eastern Asia. The Admixture View gives you a table view, which again also confirms that this person is of Eastern Asian descent. Y-DNA migration shows you the migration of the DNA given from the father. Of course, this is only for males. mtDNA migration, or mitochondrial DNA migration, shows you the migration for the mother. We'll show an interesting example later. Raw genome data gives you statistics on your raw genome data as well as your sequencing data. Genome statistics gives you a quick way to search your filtered results for any variant. This can be done through location on this picture of a chromosome, through a specific chromosome, or from this table view. The PDF symbol creates a PDF report of all of the genome annotation system reports for you in a PDF format. We at Diagnomics cannot stress enough the value of genetic information and research upon your health. We are committed to ushering in personalized medicine where one can learn whether they are at risk or not for many diseases, as well as to how they respond to certain drugs. We thank you for watching a tutorial video of our personal genome annotation system. For further information, visit our website and/or contact us. The genome annotation system does not provide medical advice, diagnosis, or treatment. Analysis and reports provided by Diagnomics are for informational purposes only and are subject to change. Consult your physician if you have questions regarding any medical condition. The results and analysis presented have not been cleared or approved by the FDA or similar government institutions. Thank you.